Spectrophotometry. Principle: ground state atoms absorb light at defined wavelengths. Line spectrum refers to the wavelengths at which an atom absorbs light. Each metal exhibits a specific line spectrum. The sample is atomized in a flame where the atoms of the metal to be quantified are maintained at ground state. Then, a beam of light from a hollow-cathode lamp, HCl, is passed through a chopper to the flame. The ground state atoms in the flame absorb the same wavelengths of light from the HCl as the atoms emit when excited. The light not absorbed by the atoms is measured as a decrease in light intensity by the detector. The detector, photomultiplier tube, will selectively read the pulsed light from the chopper that passes through the flame and will not detect any light emitted by the excited atoms when they return to ground state. The difference in the amount of light leaving the HCl and the amount of light measured by the detector is indirectly proportional to the concentration of the metal analyte in the sample. Components: hollow-cathode lamp, chopper, burner head for flame, monochromator, detector, readout device. Hollow-cathode lamp. HCl contains an anode, a cylindrical cathode made of metal being analyzed, and an inert gas such as helium or argon. Principle: applied voltage causes ionization of the gas, and these excited ions are attracted to the cathode, where they collide with the metal coating on the cathode, knocking off atoms and causing atomic electrons to become excited. When the electrons of the metal atoms from the cathode return to ground state, the characteristic light energy of that metal is emitted. Vaporized metal atoms from the sample can be found in the flame. The flame serves as the sample cuvee in this instrument. The light produced in the HCl passes through a chopper and then to the flame, and the light is absorbed by the metal in the sample. The light not absorbed will be read by the photomultiplier tube. A flameless system employs a carbon rod, graphite furnace, tantalum, or platinum to hold the sample in a chamber. The temperature is raised to vaporize the sample being analyzed. The atomized sample then absorbs the light energy from the HCl. This technique is more sensitive than the flame method. Nephelometry. Definition: nephelometry is the measurement of light scattered by a particulate solution. Generally, scattered light is measured at an angle to the incident light when small particles are involved. For large molecules, forward light scatter can be measured. The amount of scatter is directly proportional to the number and size of particles present in the solution. The sensitivity of nephelometry depends on the absence of background scatter from scratched cuvettes and particulate matter in reagents. Turbidimetry. Definition: turbidimetry measures light blocked as a decrease in the light transmitted through the solution, dependent on particle size and concentration. Turbidimetry uses a spectrophotometer for measurement, and it is limited by the photometric accuracy and sensitivity of the instrument. Molecular emission spectroscopy. Types of luminescence where excitation requires absorption of radiant energy. Fluorescence is a process where atoms absorb energy at a particular wavelength, excitation. Electrons are raised to higher energy orbitals, and the electrons release energy as they return to ground state by emitting light energy of a longer wavelength and lower energy than the exciting wavelength. The emitted light has a very short lifetime. Fluorometry. Frequently, UV light is used for excitation and is passed through a primary filter for proper wavelength selection for the analyte being measured. The excitation light is absorbed by the atoms of the analyte in solution, which causes the electrons to move to higher energy orbitals. Upon return to ground state, light is emitted from the fluorescing analyte, and that light passes through a secondary filter. The secondary filter and the detector are placed at a right angle to the light source to prevent incident light from being measured by the detector. Whereas fluorometers use filters, spectropluorometers use prisms or diffraction gratings as monochromators. Advantages: fluorometry is about 1,000 times more sensitive than absorption techniques and has increased specificity because optimal wavelengths are chosen both for absorption, excitation, and for monitoring emitted fluorescence. Limitations: changes from the established protocol that affect pH, temperature, and solvent quality. Self-absorption, quenching. Phosphorescence is the emission of light produced by certain substances after they absorb energy. It is similar to fluorescence, except that the time delay is longer, greater than 10 seconds, between absorption of radiant energy and release of energy as photons of light. Types of luminescence where excitation does require absorption of radiant energy. Chemiluminescence is the process where the chemical energy of a reaction produces excited atoms, and upon electron return to ground state, photons of light are emitted. Bioluminescence is the process where an enzyme-catalyzed chemical reaction produces light emission. For example, this may occur in the presence of the enzyme luciferase because of oxidation of the substrate luciferin. Luminometer is a generic term for the type of instrument that is used to measure chemiluminescence and bioluminescence. Chromatography. Chromatography is a technique where solutes in a sample are separated for identification based on physical differences that allow their differential distribution between a mobile phase and a stationary phase. Mobile phase may be an inert gas or a liquid. Stationary phase may be silica gel bound to the surface of a glass plate or plastic sheet. May be silica or a polymer that is coated or bonded within a column. Thin layer chromatography, TLC. TLC is a type of planar chromatography. The stationary phase may be silica gel that is coated onto a solid surface, such as a glass plate or plastic sheet. The mobile phase is a solvent, where solvent polarity should be just enough to achieve clear separation of the solutes in the sample. TLC is a technique used clinically for urine drug screening. The mobile phase moves through the stationary phase by absorption and capillary action. The solute components move at different rates because of solubility in the mobile phase and electrostatic forces of the stationary phase that retard solute movement. These two phases work together to provide solute resolution and separation. Solute will stay with the solvent front if solvent is too polar for the solute. Solute will remain at origin if solvent is insufficiently polar. Basic steps in performing TLC include sample extraction using a liquid-liquid or column technique, concentration of the extracted sample, sample application by spotting onto the silica gel plate, development of the solute in the sample using the stationary and mobile phases, solute detection using chromogenic sprays, UV light, fluorescence, and heat, and interpretation of chromatographic results utilizing RR values of solutes in comparison to aqueous standards. RR values are affected by chamber saturation, temperature, humidity, and composition of the solvent. Gas-liquid chromatography, GLC. Gas-liquid chromatograph components include a carrier gas with a flow control device to regulate the gas flow, a heated injector, chromatographic column to separate the solutes, heated column oven, detector and computer to process data and control the operation of the system. Gas-liquid chromatography is a technique used to separate volatile solutes. The sample is injected into the injector component of the instrument, where the sample is vaporized because the injector is maintained approximately 50 degrees Celsius higher than the column temperature. An inert carrier gas, mobile phase, carries the vaporized sample into the column. Carrier gases commonly used include hydrogen, helium, nitrogen, and argon. The carrier gas flow rate is critical to maintaining column efficiency and reproducibility of elution times. The types of columns, stationary phase, used are designated as packed or capillary. When the volatile solutes carried by the gas over the stationary phase of the column are eluted, the column effluent is introduced to the detector. The solutes are introduced to the detector in the order that each was eluted. The detector produces a signal for identification and quantification of the solutes. Commonly used detectors include flame ionization, thermal conductivity, electron capture, and mass spectrometer. Separation of solutes is a function of the relative differences between the vapor pressure of the solutes and the interactions of the solutes with the stationary column. The more volatile a solute, the faster it will elute from the column. The less interaction of the solute with the column, the faster it will elute. Identification of a solute is based on its retention time, and quantification is based on peak size, where the amount of solute present is proportional to the size of the peak. Area or height of the sample peak is compared to known standards. High-performance liquid chromatography, HPLC. High-performance liquid chromatograph components include solvent reservoir, S, one or more pumps to propel the solvent S, injector, chromatographic column, detector, and computer to process data and control the operation of the system. HPLC is a type of liquid chromatography where the mobile phase is a liquid that is passed over the stationary phase of the column.